Assalamu alaikum. Hope you guys are in good health. We have already discussed about the external ear and middle ear. Today we will discuss about the inner ear. Before moving towards the inner ear, I will tell you about some basic terminologies, which is the first is the frequency. Frequency, it is measured in hertz or cycles per second. It basically tells about how much waves are passing through a certain point per unit time. And if less waves are passing, we call it as low frequency. If more waves are passing, we call it as high frequency. As you can see here in the picture, this is a low frequency sound wave and this is the high frequency sound wave. Human ear can listen to the frequency of 20 to 20,000 hertz. Beyond this frequency, the human ear cannot perceive the voice. Now next is the pitch. The pitch is just like the frequency. The only difference is that the frequency is something objective while the pitch is something subjective. The pitch is what your ear perceives basically. If the sound is high pitch, it means it is also high frequency. If it is a low frequency, it is also the low pitch. Now the last one is the intensity. Intensity basically measures the loudness of the sound. The more is the intensity, the louder is the sound, and vice versa. You can see in the second picture, this is a low intensity wave. It means it will be less louder. And this is the high intensity wave. The intensity is basically measured by the amplitude of the wave. Here you can see it is of higher amplitude and this one is of lower amplitude. So high intensity sound will be louder and will be of higher amplitude. Okay, this is the inner ear. We have already discussed external, middle. Now the inner ear, I have told you we will discuss about the cochlea. We won't discuss about the vestibular apparatus because it is related with the equilibrium. While we are discussing basically here is the hearing. So cochlea is basically a coiled tubule and it is filled with the fluid. This is a cross section of that coiled tube. It is divided into the three parts. This one is known as the scala vestibuli, this one is known as the scala media, and this one is known as the scala tympani. The scala vestibuli and the scala media, they are separated by a very thin membrane known as the Reisner's membrane. This membrane is so thin that if there is a vibration or effect of sound in the scala media, it is perceived exactly the same in the scala vestibuli. So functionally they act as a same compartment. Though they have a membrane between them that is only to separate the fluid which is present in the scala vestibuli and which is present in the scala media. The fluid which is present in the scala vestibuli and the scala tympani is basically same because they both are connected with each other, while the fluid which is present in the scala media is different. This fluid is known as the endolymph, while that in the scala vestibuli and scala tympani it is known as the perilymph. Okay, we have discussed about the sound wave till the stapes foot plate which is basically attached to the oval window. Whenever the sound waves reaches to the stapes foot plate, it causes the bulging of the foot plate towards the oval window. And then when it is bulged at this cochlea, it is basically a closed compartment. So what happens, the sound waves, whenever it enters or it bulges the oval window towards interior, it basically causes the sound wave to travel and it causes the membranes inside the scala media to vibrate according to the sound wave. Then the sound wave, it moves back towards another opening which is known as a round window, and the round window bulges out again towards the middle ear. Here is another picture of scala vestibuli, scala media and the scala tympani. Now as you can see, the membrane which is separating scala media and the scala tympani is known as the basilar membrane. This one was the Reisner membrane and this is known as the basilar membrane. Now the basilar membrane, it is basically the membrane is connected with the a nerve which is known as the cochlear nerve, and it leads to a ganglion which is known as the spiral ganglion. The bony portion of the cochlea is known as the modulus. Here is another picture which is basically showing the three ossicles, and they are footplate of the stapes which is attached with the oval window. Now here again this coil tube which is basically showing in a straightened or stretched form. The area or the part of the cochlea which is near the oval window is known as the base. This one is the base, while the area which is away from the oval window is known as the apex. Now the two fluids or the two compartments, scala vestibuli and the scala tympani, they basically are connected with each other at the apex at a point which is known as the helicotrema. I repeat it, the part of the cochlea which is towards or which is near the oval window is known as the base, while the opposite part is known as the apex. Both cavities, scala vestibuli and scala tympani, they have the same fluid and they are connected at a point at the apex which is known as the helicotrema. So whenever a sound wave enters into it, it basically causes the vibration of the, as in the functionally scala vestibuli and scala media is the membrane between those is very thin, so the vibration is perceived by both, and then the sound wave travels and then comes back through the scala tympani and the round window, and there is a bulging of the round window towards the middle ear. Here is another picture, as this is a cross-sectional view, so that's why you cannot see the oval window. You can see the oval window here, and here the sound wave enters and it travels and then it goes back, causes the bulging of the round window. This is again the helicotrema, which is a part of the cochlear labyrinth where the scala tympani and scala vestibuli meet. Now we move to the basic or major component which causes the perception of the sound. It is known as the basilar membrane. As we have already discussed, it is a fibrous membrane that separates the scala media from the scala tympani. Now it contains 20 to 30,000 basilar fiber that project from the bony center of the cochlea, which I have already mentioned is known as the modulus, towards the outer wall. The fibers, the basilar fibers, these are stiff and elastic that are fixed at their basal end in the bony structure of the cochlea, modulus, and the distal ends are embedded in the loose basilar membrane. Now there is an interesting feature about the length of these fibers. I explain it to you with the picture. The fibers of the base, this one is the basilar membrane. The fiber of the basilar membrane which are near to the oval window or which are at the basal end, or which are at the base, these fibers are shorter and their diameter is more, while the fibers which are present near the apex, these are long and their diameter is small, and the fiber which is present in between these 2 points, they are of intermediate diameter and intermediate length. So what we get is the stiff short fibers near the oval window of the cochlea, while the long limber fibers near the tip of the cochlea. So how is it beneficial for us? The fibers which are near the oval window, or the fibers which are thick and which are shorter, they vibrate best at the higher frequency. So higher frequency is required to vibrate these fibers, while the fibers which are near the apex, they vibrate best when there is a lower frequency. Here you can see this is the basilar membrane. The sound wave enters and it is a closed compartment. Whenever the sound wave enters, it causes the vibration of the basilar membrane. Here in this black line you can basically see the vibration of the basilar membrane due to the sound wave. As this vibration is in the medium part or in the middle part, so it means these sound waves were of medium frequency. If these sound waves were of higher frequency, they would be vibrating this point, or if these would be of lower frequency, they would be vibrating this point. This is again a picture which is explaining the same concept. This is the stapes foot plate and this is the helicotrema. It is a higher frequency sound wave, like around 10,000. This is a medium frequency. As you can see, it is a vibrating basilar membrane in the middle part, and this is of the lower frequency. Again, this picture is showing the sound wave is entering through the oval window and it is vibrating the basilar membrane according to its frequency. Now there is another interesting feature. This blue part is the basilar membrane, and these red cells are known as the hair cells. These are basically the receptor organs or the receptor cells which basically sense the vibration. I will explain these hair cells later on. Here you can see there is a special organization. The special organization, the nerve fiber which is leaving the hair cell which is near the apex, it is different, while the nerve fiber which is leaving the hair cell from the middle part and the nerve fiber which is leaving the hair cell from the basal part or the base, these are different. So there is a special organization. So these nerve fibers, the same way, they are different and they reach at the cochlear nucleus and they can reach to the brain, and the brain can perceive that if the fibers from those near the apex, they are basically getting the signals to the brain, so it means that sound is of higher frequency. While if these fibers they take the signal to the brain can identify that sound is of lower frequency, and accordingly if it is a middle part, the brain can realize the sound is of medium frequency. Again, this picture is explaining lower frequency, medium, higher, and the complex sound. Obviously, the most of the sound which we hear during normal conversation or during music, these are complex sound, so they vibrate at the different points. So by this way, we basically can our ear can in cochlea can determine the frequency of a sound wave. Nervous system detects the different sound wave frequency by determining the positions along the basilar membrane that are most stimulated. This is called the place principle. The place principle is just like the lower frequency sound will cause the more vibration near the apex, while those of the medium at the middle part or those of the higher at the near the base. So this is known as the place principle. Again, there is a special organization of the nerve fibers in the cochlear pathway all the way from the cochlea to the cerebral cortex. So specific brain neurons are activated by specific sound frequencies. Now there is a little different about the very low frequency sound from 20 to 2000 cycles per second. They send the signal as the volley. By volley we mean they give a combined effect. They amplify or they meet with they combine their signals and then they give signal to the brain. So this is known as the volley or frequency principle. Okay, now we move to the main sensory part of the cochlea, which actually causes the sensation of the or feel the sensation of the vibration or the sound wave. This is known as the organ of CORTI. Here we have already discussed the part which basically separates the scala media from the lower part is known as the basilar membrane. The basilar membrane, here this is a amplified picture. Here you can see the basilar membrane. Now there are some special tissues which are known as the rods of CORTI, and there are two cells which are known as the outer hair cell, and this is now called as the inner hair cells. Okay, what is the organ of CORTI? Organ of CORTI is the receptor organ, lies on the surface of the basilar fibers and the basilar membrane. The sensory receptors in the organ of CORTI are two specialized type of the nerve cells known as the hair cell. One is the outer hair cell. The outer hair cell is the single row of the hair cells, while the inner hair cell, these are of the single row, while the outer hair cell, these are of the 4 or 3 rows of the external hair cells. The minute hairs or stereocilia project upward from the hair cells and either touch or are embedded in a surface gel coating of the tectorial membrane. As you can see, there are very small fibers which are originating from the hair cells. These small fibers are known as hair, simply hair, or stereocilia, and they are embedded in the tectorial membrane. Here you can see they are embedded in the tectorial membrane. The outer ends of the hair cell are fixed tightly in a rigid structure composed of a flat plate called the reticular lamina, supported by triangular rods of CORTI which are attached tightly to the basilar fiber. Now these basilar fibers, rod of CORTI and the reticular lamina move as a rigid unit. Here you can see this is the reticular lamina, in which the hair cells, they basically are embedded. This reticular lamina and rods of CORTI and basilar membrane, they make a rigid structure and they move just as a unit. So whenever there are sound waves which are causing the vibration of the basilar membrane, so as the basilar membrane and the rods of CORTI and the reticular lamina, they act as a unit. So whenever a basilar membrane moves, this whole unit moves. So it means whenever the sound waves, it causes the movement or vibration of the basilar membrane, it will basically cause the movement or the vibration of the whole unit. Okay, this is a picture which is basically showing the movement. Whenever a sound wave comes, it causes the basilar fibers to move upward. Whenever the basilar fiber will move upward, it will cause the reticular lamina to move upward and inward. While opposite, if the basilar membrane moves downward, it will cause the reticular lamina to move downward and outward. So this is how they move. Now keep in mind, whenever this reticular lamina will move, as these hair cells, they are also connected with the tectorial membrane and the reticular lamina. So whenever this reticular lamina it will move, it will also affect these hair cells, which I will explain later on. Now this is a microscopic picture of a hair cell, and these are the hair or the stereocilia. The largest of them is known as the kinocilium, and the smaller one are known as the stereocilia. These are called hair, simply hair, or the stereocilia, and this is known as the hair cell. Here again, this is a picture which is showing a hair cell, and these are the stereocilia, and the largest one is known as the kinocilium. Now, how is the impulse transmitted? How do they get it? Now here you can see all these kinocilium and the stereocilium, they are basically connected with each other by very thin fibers. Here you can see this is a geometrical picture, so this is not the original fiber, but all these are basically connected with each other through the very thin fibers. So whenever they move, so it causes the movement of other fibers too. So whenever they move towards the kinocilium, the stereocilia will all the stereocilia will move towards the kinocilium, and if the kinocilium is moved towards the opposite side, all the stereocilia will also move towards the opposite side. So how the impulse is transmitted? Here I again show you the picture. I have explained it to you that whenever a sound wave comes, it causes the movement of the basilar membrane, and all these act as a unit. So as you know, the hair cells are basically embedded in this membrane. So whenever this membrane moves, it causes the movement of both stereocilia and the kinocilium. Now keep in mind, if the stereocilia they move towards the kinocilium, as you can see in the picture, if the stereocilia they move towards the kinocilium, there are the channels which are present at the tips of the stereocilia. This yellow one is known as a channel. This yellow one is known as the channel. So whenever the stereocilia they move towards the kinocilium due to a sound vibration, of course, they basically the minute connections between those they causes the opening of these channels. Whenever these channels are opened, the positive ions, here in case these are the potassium ions, they causes the entry of the potassium into the cell. When more positivity is inside the cell, it leads to the depolarization. While opposite occurs in this case, whenever the stereocilia they move away from the kinocilium. As you can see, these small connections, here the channels were opening, now these channels are basically mechanically closed. When these channels are mechanically closed, the positive ions or potassium ion won't get entered into the interior of the cell, so the interior of the cell will be more negative, which is known as the hyperpolarization. Here again a picture which is basically showing the opening of the channels. When they move towards the kinocilium, it will causes the opening of the channel, which causes the entry of the positive ion, and opposite occur in case if the stereocilia move away from the kinocilium. Okay, I hope this is the end of the lecture. So I will explain the further the transmission of impulse and the generation of the potential in the hair cell in the next lecture, inshallah.